So in this podcast, I'm going to talk about two things. I'm going to talk about the autoionization of water, and I'm going to talk about indicators, both of which are pertain directly to titrations. So when I say the autoionization of water, what I'm saying is water breaks down into its ions automatically. So when you look at regular old water, like a container of water, you're not seeing pure liquid water. What you're actually seeing is a mixture of liquid water, hydrogen ions, and hydroxide ions. So there's always a little bit of H plus and there's always a little bit of OH minus in all samples of water. Now, some people always ask me, well, then, Mr. Siegel, what's distilled water? Well, distilled water is a process by which they take out all the ions. So that is actually pure H2O liquid. Okay, now, just on an aside, the same people always ask me, can you drink distilled water? The answer is yes, it's still water. However, your body needs ions to function. You know, that goes back to biology, the whole potassium pump thing. You need potassium ions in order to transfer nutrients in and out of cells. So if you're taking in nothing but distilled water, you are going to reduce down the number of ions that are in your body because the ions will actually get attracted to the liquid water to cause it to autoionize. So that's where when you sweat and you lose salts and things like that and you have to put electrolytes back in your system, this is why. But because water dissociates automatically into its ions, it's very useful in solutions because it can act as both an acid or a base. And I'll talk about that in a second. So what we know for water is, for pure water, the concentration of the H plus is exactly equal to the concentration of OH minus, which has a molarity of 1.0 times 10 to the negative 7. Okay, so both of them have a value of 1.0 times 10 to the negative 7. Now, what we also know is the Kw for water is 1.0 times 10 to the negative 14. So this is called the equilibrium constant for water, Kw. So the W stands for water. And what we also know, and we've done this on a previous slide, is that the concentration of H plus times the concentration of OH minus is exactly equal to Kw. So the concentration of H plus times OH minus is exactly equal to 1.0 times 10 to the negative 14, which is why we are able to calculate H plus from OH minus concentrations or from pH, etc., like we did on one of those early charts. So something I just said a minute ago, and I want to come back to, is I said water can act as both an acid and a base, depending on what situation it's in. So it can act like an acid if you put a base in it, and it can act like a base if you put an acid in it. This property of it is what is called amphoteric. Okay? Amphoteric means acts as both an acid or a base, depending on what reaction it is. How do I know? Well, I got to go back to how do I identify an acid or a base in a solution. When I talked about that in the first podcast, I said you have to look to see, does the H plus get taken in or does the H plus get given up? So you can't just say water's an acid, because it's not. It depends on what other substance it's in and how that other substance is acting. Okay, there are other substances that are amphoteric too, and we'll run into those in some of our example problems, and I'll point them out as we go through. So now we've talked about titrations, and so now we need to talk about indicators. And an indicator is any substance that's used to identify the substance as either an acid or a base. So when you do the acid-base titration lab, you use phenolphthalein. And phenolphthalein is a substance that is clear normally, but when it's in the presence of a base, it turns magenta. It turns a bright pink color. The most common one is litmus paper. Litmus paper you probably used in middle school, and they're those little blue and pink strips that you dip into liquids and they change color. So blue litmus paper turns pink when exposed to an acid. So if the pH is is about 5 or less, the blue litmus paper is going to change color. If your pH is 8 or higher, then you use the red litmus paper, where it's kind of pinkish, but it's called red litmus paper, and that is going to turn blue when exposed to a base. The other type of strip you might have used is what is called a Hydrogen strip, and a Hydrogen strip is like it's It's kind of an orangey yellow color. And when you dip it into a solution, it changes color, different colors depending on what pH it is. So it turns like red if the pH is less than 2. It turns green if the pH is greater than 10. It changes slightly reddish if the pH is like 6. So this hydrogen strips is great when you don't know the pH. If you don't know if it's an acid or base and you're trying to identify what pH range it's in. Blue and red litmus paper are great for just identifying acids versus bases. But notice there's a gap. These do not cover all pHs. There's a gap from 5 to 8. So if you have a pH of 6, well, technically that's acidic, but it won't show up. It won't change either of the litmus papers. But the hydrogen strip will change color. Then for actual chemical reactions, like I said, we're going to use phenolphthalein. And phenolphthalein normally is colorless, but when the pH turns about 8, it turns pink. So it's a great way to identify the presence of a base, and it gets darker the higher the pH goes. So when you're right around that 8 mark, it's a very, very, very pale pink. But if you get up near 11 or 12, it's a really deep magenta color, a very, very brilliant pinkish red. And there are tons of other indicators like methyl orange and Congo red and bromethymol blue and brome cresol green, and there's any number of different indicators that you can use, and they all show you acids or bases at different pH ranges. You pick the indicator that works best for your reaction. Okay, so you always pick an indicator that's best for your reaction. And I mentioned that when I talked about when I was talking about the titrations. I made a comment on the titration curve, and I said you want your indicator to change color somewhere in the range of the equivalence point, so somewhere where the two reactions are being neutralized. Sometimes that might be a low pH, and when we talk about weak substances on a later podcast, we'll talk about, okay, is the pH going to be 7 when it reacts? No, it's going to be 4. Oh, well, if the pH is going to be 4, I don't want an indicator that changes color at 8 because it'll be way too late. So indicators are it's a science. You just look at the chart. They actually have a chart of indicators. You look to see, okay, it's going to be yellow at this pH, green at this pH. Perfect, that's what I want. And that's how you pick an indicator.